All right, so now let's talk about hydrogen NMR. Hydrogen NMR is incredibly similar to carbon NMR, but now we have a last step here, which is we have to worry about the multiplicity of these peaks or the splitting of these peaks. So this is an example of a hydrogen spectrum. Notice that we still have number of distinct signals. In this case, we're going to talk about these clusters of closely aligned peaks. So this would be 1, and this would be 2. And they would correspond to the number of chemically distinct groups of, in this case, hydrogens that we have. And so in this molecule of propane, right, we have these 2 hydrogens here that are in one chemically distinct environment. And then this group of 6 is in another chemically distinct environment. So again, we still have that number of signals that correspond to the number of peaks that we get. But just note that we're going to have these peaks are going to be like split. This is, our methyl group peak has been split into this 3 trident looking thing. We'll talk about the splitting here in a second. But again, number of peaks, in this case, we have a group of the yellow and the group of the green. They correspond to the number of chemically distinct hydrogens that we would get. Just like before, the location where they fall on this chemical shift spectrum will tell us a little bit about the environment of those hydrogens. And then the intensity or the integration value will also tell us a little bit about our molecule, specifically how many hydrogens correspond to a particular set of peaks. And then the multiplicity, which we'll talk about in a second, but what you want to have in your mind is that it corresponds to the number of neighbors. So the chemical shift. So what you'll see that the scale is much, much smaller than it was for carbon. In carbon, we had chemical shifts all the way up to like 220 ppm. Here, the max is going to be like 12. Okay, so we're a much smaller signal. Nonetheless, again, it just gives you an idea. So the more electronegative an atom that carbon is bound to, the more of a downfield shift you will see, right? So fluorine being the most electronegative compared to chlorine, bromine, iodine, you see even more of that downfield shift occurring. And then the more of a particular atom you have, that will also influence it, right? So chlorine, 1 chlorine would be 3, 2 would be 5. When you have 3 chlorines, you're all the way down to 7. Okay, so electronegativity, an electronegative atom bound to a carbon will influence, shift downfield the absorbance signal for the hydrogens that are bound to that carbon. The other one that's pretty consequential, more consequential with hydrogens is the difference between a methyl, which is a carbon bound to 3 hydrogens. That's going to be the lowest shift, compared to what's called a methylene, which is a carbon bound to 2 hydrogens, and a methine, which is a carbon only bound to 1 hydrogen. All right, so you could also think about this as primary, secondary, and tertiary carbons, specifically talking about the hydrogen. But notice that basically the higher the degree of substitution, the more it's shifted downfield. So your methyl groups you expect to be the closest upfield. All right, and then just looking at the chemical shift for different functional groups. So again, all those sort of alkyl groups, they're still, even though we talked about how it goes primary, secondary, tertiary, nonetheless, they're all still pretty far upfield, our alkyl groups. You get a little bit more of a shift if it's next to a double bond or if it's directly attached to an sp hybridized carbon, also a ring. I think, good to note, again, what we were talking about, the alkyl halides, how a halogen will shift it further down. The big cutoff in my mind that you want to make is kind of what happens between 0 and 4 and then above 4, because that's where it's really distinct. So above 4 is between 4 and 8 is going to correspond to sp2 hybridized carbons. So again, benzene, and again, I'm talking about the hydrogens bound to those carbons, just to be clear, but yes. So we're talking about the hydrogens that are bound to benzene rings or that are in vinylic positions. And then another huge jump comes when we're talking about either aldehydes or carboxylic acid carbons. Okay, those are also going to be distinct, and those are huge. Those are going to be on the opposite, far end of the spectrum. So if you see something above 10, you know it either corresponds to an aldehyde and a carboxylic acid. One other thing that I would note about a alcohol hydrogen. So this particular group here, first of all, there's no splitting. And again, we'll talk about splitting here in a second, but they'll always be singlets, always just one peak. And they tend to be a little bit broader than most of the other peaks. Okay, but again, if you're gonna, if you were gonna memorize this list, that's fantastic. If you're gonna try to just like identify a few key cutoffs, this above 4 corresponding to sp2 hybridized carbons, and then above 10 corresponding to aldehydes and carboxylic acids, that's kind of the most important marker in my mind. All right, so now the multiplicity, or the splitting that we see. So when different from our carbon NMR, which only had these nice distinct sharp peaks, for hydrogen NMR, we get this like, our peak is split into a few other ones, okay? If it's just one all by itself, we call it a singlet. If it's split in 2, it's a doublet. 3 is a triplet. 4, quartet. 5 is a quintet. 6 is a sextet. 7 is a septet. Okay, so that's just the splitting of each of those peaks. Each one of these guys, so if you saw that, that would still correspond to one chemically distinct hydrogen, but that peak has been split, right? So each one of the, you see a spectrum that has a bunch of these on it. Each group only corresponds to one chemically distinct set of hydrogens. But let's see what the splitting actually means. Why do they split? So when you think splitting or multiplicity, you think the number of neighbors. Okay, that's the information that this is telling you. So a singlet and the number of neighbors is equal to the splitting minus 1. So if we have a singlet, that splitting number would be 1 minus 1. So this would correspond to, in terms of number of neighbors. And this is how many neighboring hydrogens. So 0 neighboring hydrogens for a singlet. A doublet, on the other hand, would correspond to 1 neighboring hydrogen. A triplet would be 2 neighboring hydrogens. A quartet would be 3 neighboring hydrogens. Quintet, 4, sextet, 5, septet, 6. Okay. So what the heck do I mean by that? Well, let's take a look. At something like this. All right, so notice that there are 3 compounds in this, or 3 carbons in this aldehyde. Well, I lied. Actually, let's do this one. Let's do this ketone. Okay, I have 4 carbons, and 3 of them, well, actually all 4 of them are chemically distinct, but only 3 of them have hydrogens, right? So I would expect in my H1 NMR signal to get 3 different sets of peaks because of my 3 different sets of chemically distinct hydrogens. My green peak well, first of all, we'll have an integration of 3, but in terms of the splitting, since these 3 hydrogens have 2 neighbors, meaning that there are 2 hydrogens on the neighboring carbon, we would expect this peak to be a triplet. Okay, so again, it has everything to do with the number of neighbors. There are 2 hydrogens on the neighboring carbon, leading to a triplet. If we want to take a look at my group of yellow, my yellow peak here, this yellow carbon has 2 hydrogens associated with it, right? So the integration of that peak will be 2. But now this carbon, if I look on the next door neighbor, it's bound to 3 hydrogens. So that would be a quartet that we would expect for this peak. And then likewise, our last one, the last carbon all the way at the end there, again, we'll have an integration of 3 because it has 3 hydrogens, but its next door neighbor is the carbonyl carbon. It doesn't have any hydrogens bound to it. So it has 0 neighbors and thus would correspond to a singlet.